Today I'm going to talk about our method, aMeta, that was recently published in Genome Biology, it can be installed. This tool can be installed from GitHub, and there are a lot of instructions, documentation, vignettes there, and this is a great team behind aMeta, and I acknowledge everyone for their hard work on aMeta development. But actually, today, so the goal of this presentation is not actually to convince you guys to use aMeta. It's up to you, of course. If you go for aMeta, it's also fine. But more importantly, I would like to concentrate on the methodology of aMeta, because for me personally, aMeta is not really a tool or a pipeline, but an approach, methodological approach. And I would consider my presentation successful if I could explain you guys how this methodology, why this methodology is important, and if I could give you some arguments, good arguments, why you should follow this methodology, then my presentation would be successful, I believe. Yes, so for me, aMeta is a method, and this method is based on some key principles, some fundamental principles. The overarching principle is to minimize the amount of false positive discoveries. That's the main goal. We were very frustrated finding too much in our data, something that didn't make sense, and we started thinking why these false positives pop up from time to time, and what we can do about it, and how technically you can minimize the amount of false positives. This is three other principles of aMeta. Yeah, so this is one of very frustrating examples of false positive discovery. And the Cyprinus carp, a common carp that sometimes can be found in soil samples, not because it's really present there, but this artifact has something to do with geoadaptive trimming. So we don't want this kind of hits. We want reliable hits. To get rid of this unfortunate false discoveries, we are using competitive mapping. So unbiased analysis is a central concept in aMeta. We do everything through competitive mapping, and sometimes we are even extreme in this sense. We try not to hypothesize anything about our data. So we are purely data-driven. We don't hypothesize that there is a certain group of organisms present in a sample. So we want to discover it in unbiased way. That's an essential step in aMeta. And for this purpose, you need to, in order to be unbiased, you need to utilize large and very diverse reference databases. So aMeta is a reference-based approach. We are not talking about assembly, de novo assembly. So I have to emphasize it from the beginning that we are talking about a reference-based approach. So we are using some sort of, we are assuming some sort of similarity between your query sequences and your reference sequences, which is a strong assumption if you think that microbes particularly, they evolve. And it's actually, at some point, it's a good question to ask how well this approach can work, and what do you expect from this approach. We are using large and diverse databases. It's a third essential principle of aMeta, because we want to be unbiased. We don't want to prioritize organisms. We don't want to guess what kind of organisms are present. We want to discover them. And to implement this concept, you need to use large and diverse databases. So finally, we are concentrating on evenness of coverage. That's a central metric in all aMeta analysis. We are not using numbers of assigned or aligned reads as a major criterion. We believe that evenness of coverage is a metric that describes a little bit better the actual data. And the actual data is, these are sequences. So I think it's important to remember that if you're doing ancient metagenomic analysis, we are working with biological sequences. But sometimes you might want to summarize these numbers of reads assigned or aligned and report your findings in this way as a number of mapped reads or genome coverage, which is sort of proportional to the number of mapped reads. So you see what you're doing. You're reducing this complex picture, a number of aligned reads in different way with different similarity. You reduce this complex picture down to just one number. That is undesirable. So we would not like to report our findings in this way. So that's why we concentrate on evenness of coverage, because it's a little bit better metric than just a number of mapped reads. Nowadays I don't see people reporting their findings in this way, just by a number of reads. But a few years ago, maybe 3-4 years ago, I remember it was COVID times, and I attended a lot of ancient DNA conferences, and I still have the screenshots. And every second presentation was actually reporting their hits in this manner, that they simply reported the numbers of assigned reads through Kraken or Malt or bow tie. You can use whatever tool. It was supposed to be an evidence of organism present in a sample, which I think it sounds very naive, and I hope, I will explain why. But I don't see that people are doing this anymore, because if you are after just an absolute number of assigned reads or mapped reads, if you use Kraken or bow tie and just get a number of mapped reads to a certain organism, even if you follow this competitive mapping approach, if you just get a number of assigned reads, you're into troubles. For example, that's a real world example. This is a Kraken 2 output from a metagenomic sample. So it's an analysis done on a metagenomic sample, and I rank all the organisms found in this sample by Kraken by Kraken 2. I rank them by their percent of mapped reads or number of mapped reads. And then what you can see if you scroll through the list, if you look through the list, on the top you will find some reasonable microbes. Why not? Then you find a dog or a wolf. That sounds interesting. There are a few thousand reads mapped to assigned to a dog or wolf. Then suddenly you probably you get worried because there is an eagle DNA and kiwi DNA, which makes very little sense. Then Ovis canadensis, possible, but you start getting worried. So it's something is wrong with my analysis. So what if I told you that it was a blank? Then it was really a blank. So this is a real world example. You can detect these guys present on a blank. And then, of course, you would say, I don't buy this. There are no eagle DNA. There is no eagle DNA on my blank. I don't believe that there is kiwi DNA on my blank. But if you just report your findings in terms of mapped or assigned reads, you're fine. I mean, this is perfectly. If you use a number of assigned reads as a criterion of organism detection, then yes, you might want to report these hits. Yeah, so to me, when I see these kind of pictures, I think about artifacts. So there is something that wrong with my analysis. So apparently, just to look at the number of mapped reads is not good enough. I probably have to go back to the sequences again. Like in a broad sense, the reason why these false positives pop up is because we often forget about the sequences themselves. Surprisingly, even experienced computational biologists, they don't always look at sequences. You can get trapped with numbers, and you can, like reducing data to numbers might be misleading. So if you see this kind of potentially false positive hits, what you want to do, you actually want to go back to the sequences and explore them in IGV and figure out that this is actually a false positive. And then once you understood that it's a false positive, you might figure out what to do about it. So this was how like hand-waving argument, how these false positives pop up, because we are often kind of inclined to run in some tool and getting numbers, and these numbers without sequences, they might be misleading. So this was about false positives. So what about the competitive mapping? Still, sometimes, like if I talk to people doing population genetics, not everyone actually thinks that competitive mapping is an important thing to do, because usually if you work, if you do human-centric analysis or host-centric analysis, if you're concentrating on a DNA of a host, when you're pretty sure that the majority of DNA comes from a single organism, you probably don't need to bother much about competitive mapping. But it's not the case for people working with maybe environmental and sedimentary ancient DNA. It's really important. There is no host there. You have to be more unbiased, and you have to do this competitive mapping. But anyway, if you're doing pop gene analysis, for example, you have a mammoth tusk and you extract DNA and you sequence it, and you align it to an elephant reference genome. That's good. If you're a good analyst, you might want to think that, okay, maybe I can assume that some of those reads align to the reference genome. They're not actually mammoth reads. They come from something else, from human, for example. So we humans dropped our DNA. So to fix this bias, potential bias, you want to actually concatenate your elephant reference genome with a human reference genome in order to disentangle the reads that come from human and mammoth to make your analysis more accurate. And then even if you are even better analyst, you might want to think that maybe there are more than two organisms present, or two types of organisms present in my sample. It makes sense to assume that many of these reads can be microbial. So in reality, we might have this horrible mixture of sequences coming from different organisms. And then what you...
Petrus portion, there's no pathology. Did you want me to do anything?
Sorry, was there a question? I heard someone speaking. Sorry, I didn't hear clearly. So I will continue, and then you can ask me in a while if it's okay. So in order, if you think that there are microbes there as well, so you might want to actually concatenate elephant reference genome with human reference genome and with a bunch of microbial reference genomes. So in order to make your analysis more accurate, in order to disentangle reads coming from different organisms more reliably. So, and this is what is called competitive mapping. You simply give reads a choice to be aligned to whatever reference is most similar to the query sequence. And the competitive mapping is, I would argue, it's an absolutely central concept in ancient metagenomics. But also, I hope you guys, you see what I'm doing here. When I concatenate different reference genomes, you can say I'm increasing the database size. So by database, I mean simply the number of references included in my analysis. And if I want to have a good analysis, if I want to really clean my mammoth reads from the contaminants, I want to actually include as many reference genomes in my database as possible. So yeah, I'm increasing the size of the database. And here we come to the next point, why using large and diverse databases is important. So on the previous slide, it was a little bit a cartoon. It was a hand-waving demonstration why competitive mapping and large databases are important. But here it's more a quantitative analysis, what we have done in the aMeta paper. So we actually had a bunch of simulated samples, and we knew the ground truth. So it was ancient metagenomic samples. We knew the ground truth. We knew what microbes are present there. And we tried to reconstruct these microbes. We tried to discover the microbes with Kraken unique using databases of different sizes. So on the x-axis here, what I'm plotting, it's a database size. We built a few different databases, and we thought we wanted to check how much can be, how well we can reconstruct the ground truth using a database of different size. And on the y-axis, what I'm plotting is an overlap. So this Jacquard similarity is simply an intersection over union. It's an overlap. You can think about it as an overlap between your reconstructed microbiome and ground truth microbiome. So the higher Jacquard similarity, the better you reconstruct the simulated microbiome. And it turns out that that larger and more diverse databases, they seem to give a better overlap between your reconstructed microbiome and your ground truth microbiome. This is sort of a more quantitative quantification of what I said previously about the importance of large and diverse databases. So finally, the evenness of coverage. So why we pay so much attention to evenness of coverage. So what is evenness of coverage? So here I demonstrate two scenarios of read mapping to a reference. So those are DNA sequences that are mapped to reference genome. And in both cases, in the cases A and B, I use 4 reads. So the number of reads is identical between A and B. And moreover, if you compute the coverage, so what is typically people refer to as coverage, then like according to a definition, you just take a number of reads, which is 4, you multiply by the read lengths and divide by the length of the genome. So in this case, the length of reference genome was 4 times length of 1 read. And then you can get 1x coverage, which is in both cases. You get 1x coverage for the situation A and for the situation B. But you see that these pictures, A and B, they're very different. The reads the map in a dramatically different way. So if you were to report just a number of reads, those two situations are equivalent. But I would say that in this case, when the reads map to just one region, one locus, which is potentially conserved region, and these reads, what I can hypothesize that these reads, they actually originate from another organism, homologous organism, and they just map to this conserved region. That's why I'm saying that in the situation A, I haven't detected a microbe, while in the situation B, there are good reasons to believe that the microbe was detected, because I cover more or less the whole genome, the whole reference genome. So those are four fundamental concepts behind aMeter, and it turns out they're not present everywhere, and they're not people don't use them always. I think all of these concepts, they're more or less familiar to all of you if you're sort of experienced, if you have been in the field for some time. But I don't think there are plenty of tools that actually implement all these concepts. You can ask, what is wrong about HOPES? HOPES is a very nice software, which was a baseline for aMeter. So we started with having HOPES as a baseline. So we wanted to, we realized some limitations of HOPES and try to develop this further to overcome these limitations. Yes, otherwise HOPES is a nice tool, and it answers two important questions for ancient metagenomics. Who is there and how ancient? That's pretty much what you want to know about your samples. But it has severe limitations, as I said. So people who ran HOPES and MALT, and I've been running them for years, they know that HOPES and MALT are very resource demanding. So you really need a lot of terabytes of RAM. And since not this many terabytes of RAM are not available in many labs, so what you typically do, you to compromise this, you actually work with databases of limited size. So you can't afford large databases. And remember, the large database is important for aMeter. We actually advocate for using large databases. While HOPES and MALT are feasible to run for, if you want to do unbiased analysis, it's feasible to do it only with limited size databases. And HOPES doesn't have evenness of coverage as a filter. So you can sort of tweak and extract it somehow and hack the codes, but it's not sort of delivered by the pipeline, by HOPES pipeline by default, and it's not implemented as a quality metric in HOPES. And so if you sort of ignore this limitations and work with limited size databases and ignore evenness of coverage, just to reiterate, I know that I'm saying the same thing again, but from different angles. So I think it's important to sort of a little bit reiterate it and say that if you think that large databases are not important and evenness of coverage is not super important, then I encourage you to make this simple experiment. I think everyone can reproduce it for their favorite microbe. So you can take in my case, I took a modern infant stool metagenomic sample from Broad Institute. So it was an individual who unlikely had plague. And then if you take this metagenomic sample and just map it to Yersinia pestis reference genome alone, you discover tons of reads that map to Yersinia pestis, like 20,000 or 20,000 reads. They map uniquely to Yersinia pestis because they don't have a choice. So since there is only one reference genome, you provided only one reference genome, some metagenomic reads will happily map to Yersinia pestis reference genome. And then what you can do here, you can add 10 random bacteria from NCBI-RefSeq, 100 random bacteria, 1,000 random bacteria, etc. So every time you add a portion of microbes to your database, you get this number of reads mapped uniquely to Yersinia pestis down. You get this number down. So finally, when you include, and I hope you see what I'm doing here, I'm increasing the database size. So basically adding microbes to my database, microbes other than Yersinia to my database, I'm increasing the database size. And finally, I kind of reproduce the ground truth. So I don't think there was plague in this sample, in this individual. And I actually want to have 0 reads mapped to Yersinia pestis. But still, even if you include all RefSeq microbes from NCBI-RefSeq, there will be still 20 reads mapped uniquely to Yersinia pestis, while they were given a choice to map to something else, to some other microbe. But there are still 20 20 reads mapped uniquely to Yersinia pestis. And then sometimes I heard from many people that sometimes even 5 mapped reads are considered as a good criterion for a pathogen or microbe present in a sample. So think about this. Here 20 reads can map by chance, I would say, to Yersinia pestis reference genome, while we used basically all other RefSec microbes. This contradiction, this actually paradox about mapping reads mapping to Yersinia pestis, you could have actually avoided this from the very beginning. You would have figured out that this was a false positive hit from the very beginning if you explored how the reads were mapped. So again, here on the y-axis, I'm actually using the number of mapped reads, and which is I'm saying is not a good metric. So instead, if you here in this step before adding other microbes, actually, if you would have explored how the reads are mapping, you could have figured out that this was a false positive hit. So from this exercise, what we have learned is that a way to avoid false positives in our analysis. From this exercise, I would say you can increase your database size, and then you automatically get this number of reads mapped to Yersinia pestis, which is a false positive. You get this number down almost 0, so you can figure out that this was a false positive hit in this way, just by simply increasing the database size. Or you can use evenness of coverage as a criterion, and then immediately, even for small databases, you would have said there is no Yersinia in my sample because the coverage doesn't look satisfactory. Of course, you can't inspect alignments in IGV for all microbes, all samples. That would be not possible. But at least the evenness of coverage would be a good proxy for this picture that you see in your IGV. This was an introduction to kind of emphasize that we are not only talking about a tool, but an approach which kind of was not really, can be considered as novel because it was not really implemented in HOPS, that was not able to work with large databases, and there was no evenness of coverage, so you had to do it manually. So we tried to overcome these limitations of HOPS, and there were 2 major limitations. So it was the detection accuracy. So we were not happy with the detection accuracy of HOPS because of the missing evenness of coverage filter, and the high resource demands of HOPS. It was for many samples, it was not feasible to run HOPS. And this was the motivation for us to figure out this trick. So this is a kind of an overview of aMeta workflow, but the key trick is actually here in the center. So what we decided to do is that we were using Kraken Unique in order to pre-select good candidates to be followed up with molten HOPS, basically. So what we are doing here, we are using very large reference database with Kraken Unique, because Kraken Unique can be like new modifications of Kraken Unique can be run literally on a laptop, even if your database is huge. So basically here we have an unlimited database without the necessity to have a lot of RAM. So we can be as unbiased as possible here. But this unbiased classification or taxonomic classification step, it will allow us to prioritize certain group of microbes, which can be used to build a model database around them and follow up with basically HOPS. So we are not kind of abandoning hopes. We are just introducing this pre-filtering step, because we can, Crackling has a nice feature. You can filter your hits based on both depth and breadth of coverage. So it has a proxy for breadth of coverage called the number of unique k-mers. And it turns out that it's you can concentrate, after cracking it, you can concentrate on a very reliable set of microbes to be followed up with molten hopes. That was basically the idea. So you dynamically build a database for each particular project. And this is how the main deliverable from one of the main deliverables of aMeta looks like. So we also expanded this number of quality metrics of hops. If you're familiar with hops, it uses sort of added distance and gemination, basically. But we thought we add a few more, such as, for example, the evenness of coverage plot. It's very important. So this is a central filter in aMeta, and also like other metrics such as average nucleotide identity and a few more. So we basically at the end we had 8 quality metrics to build a score around. So we use 8 quality metrics to actually assess whether a hit is present, truly present, and whether it's ancient or not. So it's not just one number. It's a combination of multiple metrics. And this, as I said, we can quantify all these plots here on this slide. You can try to quantify all of them as a score. So we combine them and create this system of scores, and you get this nice output, nice overview. You quickly can see what microbes are present or absent in what sample and how ancient they are. So basically the score number 10 means that the microbe is present and ancient, and it's a red color here. But if it's a sort of yellow and the score is 5, maybe it's present, but it's a modern contamination, maybe. So so again, you don't look at one number. You actually use a combination of different quality metrics. And since we thought it was kind of the approach we used was a little bit different from hops, we decided to compare aMeta and hops just to see whether it makes sense to do it in aMeta way, whether it gives you better results or not. And we checked it on simulated ground truth. So we simulated a number of samples, and this is sort of the ground truth. So we knew the fractions of different microbes in each sample, present in each sample, and you can binarize this ground truth on the left. You can binarize it and display it in this way. So red means present, blue means absent. So you kind of have this a map of ground truth map. So those microbes are present, okay, here it's only presence or absence. So to compare aMeta versus hops, we did the comparison using 2 errors. So we checked the detection error. So we basically tried to reconstruct this ground truth using aMeta and hops. And so both of them have a lot of hits right and a lot of hits wrong. And there is some error associated with the analysis, and we split this error into detection error and authentication error. So detection error is sort of a misidentification of a microbe irrespective of ancient status. So this error is simply when you discover a microbe, doesn't matter ancient or not, but it's not present there, for example. That would be a detection error. While authentication error is when you think that the microbe is ancient or while it's not, for example. That would be authentication error. Then it's another type of error. So we try to quantify these 2 errors for both aMeta and hops. And here there are sort of complex plots that you can check in the paper. But just to make it simple and visual, this is a reconstructed heat map with aMeta on the left and HOPES on the right. So if you compare this reconstructed heat map with a ground truth heat map, of course you can count this number of cells which are blue and red and quantify it. But I think even if you are not a convolutional neural network, you can see that here there is a lot of blue stuff. So HOPES basically was missing a lot of microbes, while aMeta was a little bit better in reconstructing the ground truth. This was the detection error. And then we also checked the authentication error. So if you're familiar with HOPES, it delivers this colorful heat map, and we were inspired by this heat map, by the way, when we were constructing our aMeta scoring system. But if you sort of run HOPES for the simulated samples, it turns out that there are a number of microbes that are sort of reported to be ancient by HOPES, which they were simulated modern. So here there were a lot of bad decisions by HOPES. And we tried to quantify it again. We constructed this sensitivity versus specificity in terms of authentication error, and for quantitative comparison of aMeta and HOPES, and the area under the ROC curve was better for aMeta. So we also claim that we are a little bit better in terms of authentication, not only in terms of detection, but also in terms of authentication. And finally, the resources. I think it's an issue for many people. We don't have unlimited resources. And aMeta is not perfect, but it tries to do its best to minimize the memory usage. So at least in this, for this simulated data set, this is this is a figure showing how what was the memory used by HOPES. This purple and this orange lines on the top. This is how much memory HOPES needed, and this is the green lines were how much aMeta was using. It was twice less, and yeah. So this. Despite we actually to make long story short, I say that simply we had a huge gain in the resources as well with aMeta. And I think 40 minutes I'm done. And I would like to, so I already acknowledged all my collaborators on the ancient DNA side from the groups of Anders Götterström and Louis Dahlén. And I also want to acknowledge my colleagues from the National Bioinformatics Infrastructure Sweden. It's basically a team of around 100 bioinformaticians spread all over Sweden. And I thank you very much, guys, for your attention.
Thank you, Nikolay. So please, if you have any question, you can unmute yourself and do it yourself and speak out, or just like Abby did, you can write it in the chat. So Abby wants to know if you can speak about whether aMeta can work with other, and if so, how much control you have in the redatabase selection.
So what is adder?
Abby, if you are there, you can unmute yourself.
Abby, can you, was it spelled correctly, or maybe it's some software that I'm not familiar with? What is adder? Or is it a organism? Functional classification. No. Yeah, I think it's a question. I think I know what you mean. So basically, if you are doing modern metagenomics, they're using a lot of this de novo assembly and also following up genes instead of sort of taxonomic classification. What we are doing here is sort of taxonomic assignment. But in modern metagenomic, the dominating approach is, as far as I know, it's to concentrate, to reconstruct genes, and then using these genes to say something about functional, like they do functional annotation instead of taxonomic annotation. I hope I understood your question correctly, Abby. And this is not what aMeta is doing. And moreover, I think many steps that are standard in modern metagenomics, I don't think they should be naively used in ancient metagenomics. They have much more power in modern metagenomics. So I basically sort of everything I read and all the talks I heard about this functional classification, they did not convince me that it works well even for modern metagenomics. So it's a beautiful idea. I'm not sure that it's sort of super successful in modern metagenomics, even though it's used a lot. I agree, and I'm not in a rush to take something that works in modern metagenomics and just naively use it for ancient metagenomics, because it should be tested. So if there are reasons to believe that functional classification can give you new insights and it's better than taxonomic classification, for example, in any way, it should be quantified and it should be a sort of given some evidence. So basically, a short answer, no, aMeta is not using any functional analysis at all, because we are not there yet, I think, in ancient metagenomics. Thank God if we can say that the microbe is present, because that's a big issue. I think it's underestimated a little bit. This issue to prove actually that the microbe is present is a little bit, the difficulty is underestimated, I think, especially in ancient metagenomics. I hope that answers your question.
Hi, can I ask the next question?
Sure, yeah.
Thank you, Nikolay. I haven't used it, but I read the article. It was really nicely written. And for me, being new to this topic, it was, probably one of the best new articles to read from this field. And it's very logical to sort of follow every step. But I wonder if, so you said that the size of database matters for competitive mapping, right? And then I was wondering if the size of reference, the genome size matters. Because if you include both bacteria and viruses and maybe parasites, because I'm working with pathogens, how does that affect the result if you looked into it?
Yeah, so it's a good question. So if you're thinking about, probably there is a connection in your question with sort of analysis that people do in environmental and sedimentary ancient DNA, where people want to discover eukaryotes. Maybe they're less interested in microbes, but they want to discover animals, birds, fish, and their genomes are huge, of course. So all these filters used in aMeta by default and that we explored, so we scanned all the possible ranges, and we are pretty sure that we are using good thresholds that are adapted for microbes. But those thresholds are not optimal for eukaryotes. And yes, so the size of genome matters. So the databases become catastrophically huge, basically. For if you use an unbiased approach working with sedimentary ancient DNA, then your databases will be much larger than the ones we use in aMeta. And I believe people have to get used to this, we are moving, in my opinion, again, maybe some people disagree, but in my opinion, to do an accurate, unbiased sedimentary ancient DNA or environmental ancient DNA analysis, you have to push the limits. You have to push it becomes extreme. The database sizes become extreme. So we are talking about several terabytes of RAM, which is it's a CERN. You have to go to CERN, supercomputers. Yeah, it's unfortunately that's a reality, and I don't have a good sort of solution. So you either use this brute force approach that you simply buy a lot of RAM, or there are also sort of alternative methodological ways to fix this problem with a lack of memory. You can split your database, and it has pros and cons. The pros is that you make your analysis feasible. The cons is that it's not a competitive mapping anymore. So you don't expose your reads to all available references simultaneously. Well, you in principle, maybe a good solution would be you can still split your databases by the groups, by kingdoms, for example, but you make sure that you expose your read simultaneously to all databases, sort of not in parallel. I meant in parallel, not sequentially. So sometimes people just use one database. They process their data through this database, then plug in another database and process the rest, the leftovers. They process through a second database. So that would be a sequential way. But I believe the more important is to expose your reads in parallel to all. If you choked your database in chunks, so please make sure that you expose your reads in parallel to all databases, and you make a decision. Sometimes a read can be assigned to multiple databases, and then you have to decide. You have to look at the affinity. You have to then make a decision. So if a read is classified in invertebrate database and microbial database, for example, you have to decide whether it's an invertebrate or microbe. And you have to check the affinity. You have to really go back to the sequences to check how it maps, with what affinity, what is the similarity, how many mutations, how many mismatches, etc.
And do you take the whole genome? Or, for example, if you have multiple chromosomes, you choose some chromosomes? If it's a big genome, can you compromise by subset it and take only part of the data?
Yes, sort of that would be going back to this mitochondrial. So that people sometimes screen for what kind of organisms are present in their samples using sort of not whole genome data, but yes, sort of it's a barcoding or meta barcoding way, so that you do not use full lengths of the genome. So I think, yeah. So again, there are pros and cons. It should be so. Like in theory, of course, if you can afford, you should go full genome. But yeah, it should be quantified. It should be tested how reliable results you get from. So if there are several ways to follow, I believe, well, ideally what should be done, you take simulated data, you simulate multiple organisms present in a sedimentary ancient DNA sample, and you test all your approaches. So if you believe that, okay, you can chunk your database, you can split your database in parts, or you can use one chromosome, or you can use full genome. Try to reconstruct the simulated ground truth. So if you see that you are wrong in your conclusions, you're severely wrong in your conclusion by compromising, by using only parts of genome, then probably you should not do it. It should be all quantified.
Thank you.
You're welcome.
Does anyone have any more question?
Can I ask a question as well, please? Thank you for that great talk. It was really, really informative. I am just wondering, because I've already got a Kraken 2 database generated, how would you advise changing this to a Kraken unique to start the aMeta? Because I'm trying to look for eukaryotes, and I think when I looked at the aMeta, it starts with like a microbial starter for the Kraken unique. So I was just wondering how, or if you know how to change that over.
Yeah, so when we were deciding between Kraken unique and Kraken 2, so we decided to go with Kraken unique for a reason. So at that time, a few years ago, there was only Kraken unique that used the breadth of coverage filter. So it was not available in Kraken 2. That's why it was like a major reason for us to stick with Kraken unique. Another advantage, more recent advantage of Kraken unique is that once you build your database, you can run your analysis on a laptop, basically. That's a huge advantage. It's not the case for Kraken 2. For Kraken 2, you still might need a lot of RAM. That's the second advantage of Kraken 2, of Kraken unique. And then nowadays, I think this minimizer filter or breadth of coverage, the proxy for the breadth of coverage filter was recently, or like 1 year ago, 2 years ago, it was added to Kraken 2. Again, after a lot of testing, so I was comparing actually they claim, so the Kraken 2 developers, they claim that this minimizer filter, it should be identical to the number of unique k-mers used in Kraken unique, because it was literally imported from Kraken unique to Kraken 2. But after a lot of testing, so my testing was a little bit inconclusive. I still doubt. I'm still not fully confident that it was an equivalent of the number of unique k-mers. So what I want to say is that this breadth of coverage filter available now in Kraken 2 as well, in theory, did not convince me that it was fully equivalent to Kraken unique. Yes, just to answer your question regarding, yes, you have your Kraken 2 database built. Right now, there is no way to use this database with aMeta, unfortunately. So you have to build Kraken unique database. Unfortunately, they are very different. So Kraken 2 database is very different from Kraken 1 and very different from Kraken unique. So Kraken 1 databases, they're very similar to Kraken unique. So it's basically the same database. So Kraken unique is using Kraken 1 databases, so to say. But then when Kraken 2 paper, in the paper in this new development, they claimed that they they made Kraken much faster, and Kraken 2 is indeed very fast. And also the databases, they changed a lot between Kraken 1 and Kraken 2. The database changed a lot. If I'm not mistaken, the compromise was the accuracy. So they actually managed to reduce the database size and utilize less memory with Kraken 2. But the price was actually that the accuracy was a little bit sacrificed. So it was, to the best of my knowledge, to the best of what I revisited this paper a few times, and it's kind of written a little bit unclear. Just looking at the plots or in their figures, I can see that some accuracy was sacrificed by this modification. But they don't state it clearly in the paper. They don't say that accuracy is somehow affected, or they stated in somehow like, I still, yeah, it would be some alternative opinion. I would appreciate it, because also my colleagues, and I talked to many people who are also kind of unclear with this. Basically, we provide a few cracking unique databases. So if you have your Kraken2 database. So in order to run aMeta, you can't use this build Kraken2 database, but instead you can take our database if you want. If your Kraken2 is special in some sense, then I would advise you just to build a Kraken unique on the top, using you take these references used for Kraken2 database and you build a Kraken unique on the top. If you don't want to use our database, because we published a bunch of actually Kraken unique databases for different cases.
Okay, great, thank you. I'll have a look. I think I don't know. I speaking to my colleague about this, and she was like, "Oh no, we'll have to build one on top because it's only like microbes." So I'll have another look. But yeah, that's really helpful. Thank you.
Yeah, but just if you need any assistance in building a Kraken unique database for your particular microbes that were included in your Kraken2 database, I think I could help you. So it's not super difficult.
Great, thank you.
Let me know. Yeah.
Yeah. Okay. I'll have a go. Thank you very much.
You're welcome.
Can I have another question? If there is still time.
Sure. Yeah.
Yeah.
Absolutely.
So another question. So I never set up this Kraken analysis with Kraken database, but I was wondering because you filter so the results from Kraken unique sort of affect your database, what references you're going to choose for competitive mapping.
That's correct. Yes. So here, basically, only microbes selected on these steps are propagated to multi-step. Yes.
But other references are the same. So if there's like a hit on Yersinia pestis in Kraken unique, do you use exactly the same reference with the same ID?
Yes, I do.
Okay.
So basically, all the microbes, so the references included here, it will be the same references used here.
Yeah. Okay. Yeah, because I feel like it's very difficult, or at least it's difficult to choose reference sometimes. There are just overwhelming number of sequences. So yeah, that's probably a whole different topic, how to choose a reference, but at least I know that it's the same in both. Okay.
Yeah, so we there are two major NCBI nucleotide databases. One is NT, the other one is RefSeq, and the RefSeq is much better in terms of the quality of references. Unfortunately, it has, like talking about microbes, it has like 36 or like 40,000 microbes. And if you look at the NT, it's much bigger. So there are many more microbes, but there is also a lot of garbage there. So there are a lot of sequences of very poor quality. And then there is a choice. So do you concentrate on good quality references, but very few microbes? And it's not good for diversity. So you won't be able to discover microbes that are not in RefSeq. RefSeq sort of does not cover very well the microbial tree of life. But if you go for NT, NCBI NT, so you will have a lot of redundancy. So lots of sequences, there will be just duplicates, and which will consume a lot of RAM, but will be not very useful for the analysis. But unfortunately, you have to decide. But what you gain with NT, you get diversity. So you increase the diversity, and this is what we prioritize. That's why we thought NT was better. Yes, by the way, when we were testing, that's probably a good illustration of NT versus RefSeq. When I was talking here, so this is a full NCBI NT, and this microbial NCBI NT, and it had a sort of the microbes that we simulated, for these simulated microbes, we reconstructed them much better with NT compared to RefSeq. So here, when we used only RefSeq complete genomes, the reconstruction was not great. So you should keep in mind that diversity also matters. And that's why, yeah, so we prioritize the diversity.
Okay. Yeah. Thank you.
You're welcome.
Okay. So, I mean, I have many questions, Nikolay, but I think I will ask them to you maybe in another context, because I think we are quite already like over the time for the meeting today. So I guess if nobody has any other quick question, we can close the meeting and thank Nikolay. This was very enlightening.